Hello everyone, and welcome to today's session, Automating Workflows for Drugs of Use Testing by LC-MS, Labor Berlin's Experience. I'd like to start by introducing today's speakers. The first one is Dr. Lars Ambach. He is the forensic coordinator at Labor Berlin's Toxicology lab. He holds a PhD in chemistry and molecular science, and he has over 12 years of experience in clinical and forensic toxicology, with a strong focus in LC-MS. His key roles include coordinating forensic toxicology testing, as well as method development and validation. I will be the second speaker. My name is Gabriela Boza-Moran, and I am the team leader field marketing at Hamilton Robotics. My educational background includes a PhD in molecular and cell biology and an EMBA in management of technologies. I have more than 10 years of experience in scientific research, and one of my roles in Hamilton is to create and disseminate scientific content describing the use of Hamilton products in various application areas. At the end of the webinar, Dr. Nikolai Kraut will join us during the Q&A. He is Hamilton's LC-MS automation expert. Nikolai is the product manager for applications at Hamilton. He holds a PhD in bioanalytical chemistry and has over 10 years of academic and industry experience in analytical chemistry. In his current role, Nikolai translates customer requirements into viable products of lab automation for analytical chemistry, cell biology, and other applications. Before we begin, I would like to mention some important aspects. So first, this webinar is being recorded and it will be available for download afterwards. We want to hear from you, so please write your questions right there in the Q&A box. We will do our best to answer all your questions at the end of the webinar in the Q&A session. And if for some reason we cannot do that, we will come back to you by email to answer your questions. Now, for those of you who are joining us now, welcome, and we will begin now with the presentation. The agenda for today's webinar is as follows. So Lars will first give an overview of Labor Berlin before describing what is drugs of use and what is toxicology, what compounds are measured in clinical setting, and why that is important. And after this, Lars will describe the sample prep methods they have automated for drugs of use testing via LC-MS. I will then take over and give an overview of Hamilton and the solutions we offer in the field for the preparation of LC-MS before analysis. I will finish now with the Q&A session. And as I previously said, please write your questions as they come along, as they pop up in your head, write them in the Q&A box, and we will discuss them at the very end. I will now give remote control to Lars, who will talk about Labor Berlin.
Okay, thank you, Gabby, for the introduction. I would like to start my part of the webinar with just giving you a quick overview of Labor Berlin, the company that I work with, to give you a background in what we do and why we do it. So Labor Berlin is a company that belongs to two parent companies that are publicly owned. One of them is the Charité University Medical Center in Berlin, and the other one is Vivantes Network for Health, that is also a company owned by the city that runs several hospitals within the city of Berlin. So we have 9 different departments, and we operate on 13 different sites throughout the city of Berlin that you can see pictured here on the map. I myself work at the Campus Virchow-Klinikum, pretty much in the city center. And that is also the site of our main building that houses most of our central laboratories. Furthermore, we have 23 associated hospitals in the city of Berlin that also send samples to our labs for analysis. As I've already mentioned, we have 9 different departments that cover pretty much everything there is in clinical diagnostics. We have the Department of Laboratory Medicine and Toxicology, where I work. We have microbiology, virology, genetics, allergy diagnostics, autoimmune diagnostics, endocrinology, hematology, oncology, and an immunology department. So that is a pretty complete spectrum, I would say, in terms of services that are required in clinical diagnostics. As I've already said, we're a private company, but we're publicly owned because our owning companies are state-owned. We perform more than 60 million analyses every year that generate a turnover of more than 70 million euros each year. And our services are focused in particularly on servicing the Berlin area. We have more than 600 employees in our 9 departments. As I've already mentioned, we cover quite a big diagnostic spectrum. And of course, we operate 365 days a year, 24 hours a day, round the clock, both in performing diagnostics and providing consultations for the medical personnel on the wards that send us their samples. And also innovation and development is a big topic in our company. For example, we have an innovation fund set aside that distributes more than 750,000 euros every year into innovation projects and throughout the company. Labor Berlin is also, together with other laboratories in Potsdam, Chemnitz, and Frankfurt, the founder of a nationwide network of medical laboratories. And in this network, we care for more than 24,000 hospital beds throughout Germany. So that was it about the company. I will now move on to the probably more interesting part, which is drugs and toxicology. So I think drugs are always an interesting topic to talk about. I will start with a little bit of the basics. What is toxicology? Toxicology is the science dealing with the effects of toxicants or toxic substances on living organisms. And within that sort of general description and pretty wide field, we have the subdiscipline of analytical toxicology. So there we are concerned with applying methods of analytical chemistry to determine which substances are in a sample, in what concentration are they present, and so on. Analytical toxicology can be employed in clinical context. For example, we do therapeutic drug monitoring, where the question is whether the medication has the right concentration within the blood. Does the doses need to be increased or decreased? Is it the right dose that is given to the patient, and so on? We also do emergency samples, for example, from intoxication cases. So a patient is admitted to an emergency ward with unknown symptoms and might be intoxicated with an unknown substance. So then the job of the clinical toxicologist is to find out which substance could cause these symptoms. And we also do forensic toxicology. So that is toxicology applied in a judicial context. So it's always something to do with the law. It's part of criminal investigations. For example, there might be accidental intoxications. There might be deliberate poisonings of other people. Post-mortem toxicology deals with determining the cause of death, which is not something that we do. That's usually done by departments of forensic medicine. Yeah, and another question is also abstinence monitoring. So, for example, somebody has been convicted for driving under the influence offense. people that are currently serve prison sentences, that are on probation or that undergo therapy. Those people usually have to show that they don't take drugs anymore, and so they're part of an abstinence monitoring program. So there are different reasons to do drugs of abuse testing. As I've already mentioned, one might be an acute intoxication of an unknown cause. So, for example, there's an unconscious person and they might be unconscious because they've taken drugs or they've drank too much alcohol. And yeah, then it's our job to determine whether there's a toxicological cause for the symptoms that the patient experiences. Then you might test for drugs of abuse for clinical abstinence testing. So, for example, a patient has a health issue with the use of addictive substances, and that has caused them physiological problems, and therefore they should cease consumption. And then you do abstinence testing to determine whether they're actually not taking the substances anymore. On the other hand, there's also forensic abstinence control. So, for example, a patient that has violated narcotics laws or other laws, and they have to prove they're abstinent as part of the sentence that was given to them by a court of law. And last but not least, there's also patients undergoing therapy. For example, somebody who's dependent on heroin. They usually undergo substitution therapy with either methadone or buprenorphine. And then you do drugs of abuse testing for two reasons. The first one is to check for compliance. Are they actually taking their substitution medication? And the second reason is to exclude the consumption of other substances. So they should not take anything but their medication. So drugs of abuse testing is quite interesting because it covers a wide array of different substances and substance classes. We have opiates and opioids like morphine, codeine, methadone, tilidine, tramadol. We have benzodiazepines like diazepam, Z-drugs like zolpidem and zopiclone, cocaine, for example, and its metabolites, different amphetamines like methamphetamine or MDMA, also known as ecstasy, or some newer psychoactive substances like cathinones. Of course, the all-time classic substance is THC, the principal component in cannabis. And there's also several different other substances that we need to check for example, some medications that might also be abused by people. And to check for drugs of abuse, we employ two fundamentally different analytical approaches. So what we usually do at first is that we use screening by immunoassays. So these are usually group assays where you can screen for a whole array of related compounds at once with a single analysis in a couple of minutes. They're quite fast. They're also quite cheap. You can screen for different substances at once because the antibodies used in immunoassay will bind to a common structural backbone in these related molecules. So, for example, you can have an immunoassay for amphetamines that will detect amphetamine itself, methamphetamine, MDMA, because they all share the phenethylamine backbone. So that's what we usually do at first. Of course, because immunoassays are antibody-based, there's also the issue of cross-reactivity. So you might have compounds that are not actually drugs of abuse, but that also bind to the antibody that is used in the assay. So there's always a risk of generating a false positive result with an immunoassay, which is also the reason why an immunoassay result alone is also not admissible in court and has always to be confirmed with a specific analytical technique that is able to identify a single compound by itself. So once we have a positive result in an immunoassay, the sample is usually reanalyzed with a chromatographic technique, because these techniques are specific. We can identify the single compounds that actually cause the positive result in the immunoassay. So, for example, we use GC-MS methods, so gas chromatography coupled to mass spectrometry. We have liquid chromatography coupled to tandem mass spectrometry, and we also have liquid chromatography coupled to a time-of-flight spectrometer, where we can do exact high-resolution mass analysis. And yeah, as you can see, we do use GC-MS for things like buprenorphine, opiates, cocaine, methadone, and THC. On our LC-MS instruments, we also have methods for which we don't have any immunoassays, for example, synthetic cannabinoids, cathinones and piperazines, opioids like tilidine and tramadol, and also GHB. We don't have any immunoassays for that. So if those substances are suspected in a case, the samples are directly analyzed with the corresponding LC-MS method. And we use our QTOF to analyze for barbiturates and for benzodiazepines. Now you can also use quite a wide array of different matrices for drugs of abuse testing. So first of all, there's, of course, blood or its derivatives, serum or plasma. The detection window for most drugs of abuse in blood is a couple of hours, maybe a day if you're lucky. And yeah, the challenge with blood is also always that it's an invasive sampling process. You have to stick the patient with a needle. And yeah, so blood samples are usually used for intoxication testing. So in case of emergency samples, because they give you an overview of what's happening in the patient right now, which is what interests you if you have a person in the emergency department. We can also use dried blood spots for drugs of abuse testing. So these are less invasive than regular blood samples, because you just use a finger prick, and the capillary blood from the finger prick is then spotted onto filter paper. Detection time is, of course, the same for different substances. Then the classic matrix for drugs of abuse testing is urine, because it's very easy to get, and you have a detection window usually of a couple of days up to a week for compounds like THC, or certain benzos can even be a couple of weeks or a month. The downside is that urine samples can be quite easily manipulated, so you always have to observe the sampling process. Oral fluid is still fairly new for drugs of abuse testing, and of course I would talk about our oral fluid method a little bit more in detail later on in the presentation. Again, the detection window is a day to a couple of days. It can be challenging because analyte concentrations can be quite low. Now, if you want to do long-term testing, if you want to cover a long period of time with a single sample, hair is your matrix to go for, because with hair you can easily cover a couple of months depending on the length of the hair. So hair usually grows 1 centimeter a month. So if you have a hair sample that's 4 centimeters long and you homogenize it, you can cover a period of 4 months within a single sample. But the analysis process can be quite complex, and the interpretation is also sometimes tricky and not so straightforward. And quite a special matrix that we also analyze in our laboratory is meconium. So that's the first stool of a newborn baby. And in meconium, you can detect substances that the mother has consumed during the last trimester of her pregnancy. So that's it's usually of importance when you have parents that have a history of drug dependence, and then the authorities are debating whether they are allowed to keep custody of their child, then they usually want meconium analyzed to see whether any drugs were consumed during the pregnancy. And yeah, it's a bit of a difficult matrix because it's inhomogeneous, it's quite sticky, so it's also a bit tricky to analyze. I just want to go a little bit more in detail for two matrices, that's urine and oral fluid. As I've said, urine is the preferred matrix when it comes to drugs of abuse testing because it's very easy to sample, it's non-invasive, you have high analyte concentrations compared to blood samples, so you don't need the most sensitive instrument for that. And you have a considerably longer detection window than in blood samples. On the other hand, you also have lag time. So if you take drugs, it takes a little while till they actually show up in urine. So for example, if I would consume a drug right now, would give a urine sample 5 minutes later, that compound may not be present in the urine sample because, of course, it takes a little while until substance has gone through the bloodstream, is excreted through the kidneys and actually accumulated in urine. And yeah, depending on your fluid intake, for example, your physical condition and so on, concentrations in urine can also vary. So usually it doesn't make a whole lot of sense to do quantitative analysis in urine because the interpretation of those concentrations is not easily done. And because I've already mentioned, urine samples are also easily manipulated. So for example, you might just put in some water from the toilet if you're giving a sample to dilute the urine because you want it to show up negative as a patient. And there are, if you Google it, there are quite elaborate contraptions with bags of artificial urine that can be hidden in your pants that are then filled into the cup where you're supposed to pee. And there's just quite a lot of creativity going on there. So it's always important that a third person controls whether the sampling process was done properly. So that's, of course, a little bit a privacy issue because, yeah, you have to watch a person pee into a cup, really watch them. So that's not a comfortable situation usually for all parties involved. On the other hand, a new matrix that was requested from our customers is drugs of abuse testing in oral fluid. The advantage is that it's a non-invasive sampling process as well. You get a rinsing solution that you have to put in your mouth, then you have to rinse your mouth for 2 minutes and spit the solution back into a beaker, and from the beaker it's transferred to a tube, which then sent to our lab. So it's quite an easy sampling process. You can, like a prison ward or a medical professional can observe the sampling process without causing any privacy issues because, yeah, the patients don't have to drop their pants in front of you, to be blunt. And it's also harder to manipulate in urine because, yeah, you can't hide anything around your mouth usually. On the other hand, the diffusion of analytes from the blood to oral fluid varies depending on the compound. So there's no direct correlation between blood concentrations and oral fluid concentrations. So usually, yeah, it's also not that straightforward to interpret. So in the next part of the presentation, I will talk about automation in our lab and how we use that to also develop a method for the detection of drugs of abuse in oral fluid samples. So here you can see the deck layout of the Hamilton Microlab Starlet that we use for our automated sample preparation. We have different carriers that we use. So we have pipette tips that are used by the robot arm for pipetting. We have a carrier for deep well plates that I use for mixing, sample preparation, sample collection. There's a carrier that carries different reagent troughs. So if you need any reagents in your sample preparation process, these can be placed here. Of course, there are carriers that are used for the sample tubes to be loaded into the robot. We have a lid park position for a lid for our deep well plates. There's also a cooling module. So at the end of the sample preparation process, the well plate with the finished extracts is placed in a cooling module until somebody is there to actually unload the samples and take them to the analytical instrument. We have a heater shaker that we can use for mixing and also for incubation reactions. There is a vacuum station, which we use for solid phase extraction methods to suck the liquid through the SPE cartridges. There's also centrifuge that can be used for well plates. And last but not least, we also have an autoloader with a barcode reader. So that barcode reader can also scan the labels on the patient samples. So we have sample tracking within the sample preparation process. We've developed already a few different methods in our lab. That was mostly done by colleagues of mine. So you can see we also used several different sample preparation processes. So we have a couple of methods that use protein precipitation. We have a method that uses hydrolysis. And the method that I want to talk a bit more in detail now, the drug screening in oral fluid, this uses solid phase extraction for sample preparation. So let's move on to that method. So first of all, the sample has, of course, to be collected. This is usually done not in our lab, but at our customer's place. So our oral fluid samples mostly come from prisons around Berlin, and they want to move away from traditional urine drug screening to oral fluid, exactly because of the privacy issues that I mentioned. Because yeah, you can imagine that's not a particularly nice situation to be in, especially if you consider what kind of people are usually in these prison wards. And collecting oral fluid is much more simple, much more convenient in that situation. So what we have is a tube with a yellow-colored extraction solution that has to be put in the mouth by the patient, and they have to rinse their mouth for 2 minutes with that. Then that solution together with is then, of course, mixed in the mouth with saliva and collected into a beaker. And from the beaker, it's transferred into a vacuette tube with an adapter that is integrated into the lid for the beaker. And these transfer tubes are then sent to our lab for analysis. So once they arrive at our lab, there's 3 different things, 3 different analyses that we perform on these samples. So the first one is checking the alpha amylase activity. Alpha amylase is an enzyme that's present in saliva in high concentrations. So that is to check whether we actually received an oral fluid sample. So to prevent somebody from just sending us the pure rinsing solution, for example, we check whether the amylase activity is high enough in the sample. Then we also determine the ratio of actual oral fluid to the rinsing solution. which is done on a photometer by measuring the absorption of tartrazine, which is the compound that actually makes the rinsing solution yellow. And last but not least, the biggest part is the sample preparation for the LC-MS analysis, where we check for the actual drugs of abuse. So that's done with a multi-step SPE protocol, and in the LC-MS method, we then check currently for more than 65 analytes in a 4.5-minute runtime. So I will explain the sample preparation process a little bit more in detail now, so that you can see what kind of different steps you can actually do with the Hamilton instruments. So first of all, samples, reagents, consumables are all loaded onto their respective carriers into the instrument. Then the samples are checked and verified via automated barcode reading. And then for the first part, for the alpha amylase activity test, 10 microliters of each sample are pipetted into a new 5 milliliter plastic test tube. There they are diluted with 1 in 100 with a special dilution buffer. And then these tubes are taken out and are sent downstairs to another department where they are checked on an immunoanalyzer for their alpha amylase activity. And the second part, the determining of the ratio of oral fluid to the rinsing solution, we simply aliquot 200 microliters out of each sample tube onto a 96 microplate well. And once that's done, that microplate is taken out, taken to a photometer with a plate reader. And there we check the absorption and calculate the actual ratio of oral fluid to rinsing solution. And now the most complex part, that is the SPE method as a sample preparation for the LC-MS analysis. There we use 200 microliters of sample. Those are mixed with 15 microliters of an internal standard mix in a 96 deep well plate well. Once they are pipetted into the well, the deep well plate is shaken for 10 seconds to mix everything. And then the samples are transferred to a microelution SPE plate. Here we use a Phenomenex polymeric strong cation exchange material to do solid phase extraction. And once the samples are transferred to the plate, they're first washed with 100 microliters of a 100 millimolar sodium acetate buffer. In a second wash step, they are washed with 100 microliters of 30% methanol. Then the SPE plate is dried with the vacuum station. And in the last step, we elute with 2 times 50 microliters of 2.5% ammonia in cetonitrile. And these, the eluate is then collected into another deep well plate, which is then taken to the autosampler of our LC-MS system. I will just very briefly mention some facts about our LC-MS method. So we use a Shimatsu system for that. So it's an HPLC system coupled to a Shimatsu LC-MS 8060 instrument. We run a 4.5-minute gradient, and we are able to detect more than 65 analytes within that runtime. We use isotope labeled internal standards for almost every analyte. So we have about the same amount of different internal standards as we have analytes. So that, each analyte almost has their individual internal standard. And for each analyte, we also measure 5 different MRMs as opposed to the regular 2 2 to 3 that I usually do with MRM methods. And we plot these, the responses for each MRM transition for an analyte, sort of like a product ion spectrum to generate what we call an MRM spectrum, which is then matched against a library search. So this is a Shimatsu specific technique as far as I'm aware. Yeah, that gives you quite a high identification power because yeah, you measure 5 different signals per analyte and put them in relation to each other. So to summarize why we use automated sample preparation for this method and what kind of benefits it gives to us. Well, first of all, I have to say, without automating the sample preparation process, it would not have been possible for us to even start with oral fluid testing for drugs of abuse, because our customers have projected quite a high number of samples for the future. So they say they want to send us 100 to 150 samples per day. So if you have to prepare that with solid phase extraction, it's only possible for us through automation. If we would have to do it manually, we would have already had to say at the start of the project, we can't do it that way. Another big advantage is that the method that we developed is a walk-away method. So once you start the process on the instrument, it does everything automatically. You can walk away, literally do something else. You free up time for other tasks by using automation. In our experience, that's especially useful for large batches and analyses that can be planned in advance. So if you know we get 100 samples every day, that's fine. It's a little bit more difficult if you have like a single sample for an emergency case where you have a short turnaround time. That's something that we still do manually. But for things like that, that are large amount of samples that can be planned in advance, automation is really, really good. Then we have sample tracing via the barcode reading. So we always know which samples are processed, and the Hamilton instrument also generates mapping files. So you have a correlation between the barcode on the sample tube and the position of that sample on the well plate. That is, of course, documented. And you can use these mapping files directly to generate analysis batches in the software of the photometer and of the LC-MS system. So you don't have to type it manually anymore. It's just a simple copy and paste process from the mapping file into the instrument software. So that is quite handy. And that, of course, also prevents typing errors or any sort of mismatches. As I've also shown you, the deck layout that we have is a generic deck layout that gives you quite a bit of flexibility for different workflows. As I've mentioned, you can do protein precipitation, you can do liquid-liquid extraction, you can do SPE, you can do simple aliquotations, you can do incubation reactions, and so on. So it's a flexible instrument to use. And of course, with automation, also reproducibility increases because you eliminate manual errors, sample mix-ups, and so on. And you also have precision pipetting by automatic pipetting. So this concludes my part of the presentation. If you have any questions, write them in the Q&A box on the bottom, and we will get back to those questions at the end of the webinar. Thank you very much for your attention. And with that, I give back to Gabby. Thank you.
Thank you, Lars. This was very, very interesting. I will now move on to give a brief overview of Hamilton and about the other solutions that we have for LC-MS sample prep. A brief introduction of Hamilton. So it's a private group founded in 1947. Today we have 2 headquarters. So the laser points, we have one in Reno, Nevada in the US, and one in Bonaduz, Switzerland. And we have a total of 5 manufacturing sites, which include another site in the US in Franklin, another site in Switzerland in Domat M, and a site in Romania. Altogether, the company employs more than 3,000 employees around the world. And that includes also the 7 subsidiaries offices that we have throughout Europe and through China. Hamilton is divided into 5 independent business units: robotics, storage, process analytics, laboratory analytics, and medical. And in this webinar, we are focusing on the products of robotics. So robotics is specialized in the manufacturing of liquid handling instruments, both for OEM and also for direct sales. And here's where I would like to give a little bit more focus. Hamilton Robotics has 4 platforms for automatic liquid handling. So one, and our newest one, is our Microlab Vantage line, which is the next generation of liquid handling automation. Our classic one is a Microlab Star line. We also have 2 smaller ones, the Microlab Nimbus line and the Microlab Prep. And this particular webinar, we are focusing in the Microlab Star line, or this is the one that was described by Lars. And we're going to go a little more in detail in the modules that we can offer insight for the sample prep for LC-MS. In terms of applications, our solutions are used in the automation of workflows in various research areas. That includes genomics, analytical chemistry, proteomics, cellomics, compound handling, but in general, it's anything that requires basically liquid handling. And we have different modules that could be included in the platform then to do other types, more elaborate workflows. And in this particular webinar, we are focusing in analytical chemistry and more specifically in LC-MS. Now, what are solutions for LC-MS? So we offer 2 workstations designed specifically for LC-MS sample prep. We have the Dual Master and the Chrome System Master. And both solutions are based on our Microlab Starlet. They have predefined hardware and software packages. If we go a little more in detail, the Chrome System and Master Dual Star, they differ in terms of the throughput and the level of regulatory compliance. The Dual Master is research use only, and it is able to process up to 80 samples at once. And the Chrome System is an IVDD compliant solution processing up to 288 samples per batch, although we do not recommend running more than 192 samples at the same time. It should be noticed that the Chrome System Master is currently sold as a complete IVD certified solution in combination with the Chrome System kits, Mastox immunosuppressants, Mass Chrome 25OH vitamin D. In terms of available methods for the Dual Master, so I think Lars presented very well several of the methods. We actually have collaborated a lot with Labo Berlin to develop these methods. And he already talked about the oral fluid method for analyzing drugs of use testing in oral fluid samples, antibiotics, antidepressants. So we know that LC-MS is a critical tool for the analysis of samples in the areas of clinical, forensic, and toxicology. And we collaborate with leaders in the field, such as Consistent, Recipe, Waters, Thermo Fisher, to make sure that our solutions are compatible with their workflows and with their instruments. So as I said, our platform can be equipped with a lot of different modules and tools to do more complex workflows. And here I would like to show some of them. So they can be equipped with various different modules. And here we have the first one is the flip tubes, which can be used for close reactions. And the flip tube provides an alternative to using plates. And it can also be, we can equip them with flip tubes tool set that basically automates the opening and the closing of the flip tubes while preventing the aerosol formation. We also have our MP2 square, which is short for Monitor Multiflow Positive Pressure Evaporative Extraction. So basically it's for solid phase extraction with positive pressure. We also have a vacuum-based SPE module, which is the one that Lars was talking about for customers that prefer this solution. Another module is our centrifuge. Also, Lars showed it on the deck, usually integrated on one side of the deck. We have our ODTC, or on-deck thermal cycle, which we use for incubations between 4 and 99 degrees. Our punching module, which we use for card punching, and it recognizes possibly stained samples and also negatively stained samples. And then it makes punches of 1.2, 2, 3, and 6 milliliters directly into the collection plate. We also have the APH model for pH measurement, a sealer for adhesive plate sealing with optical clear or pierce foil. We have piercing tips. And we have the two last solutions that I'm showing here for integration. They are based on partnership with other companies that we see here on the left-hand side. First, we have the specialized tips. And here in this case, we are showing the tips in a 384 head. And with these, basically, with these tips, samples can be filtered, and with other samples can undergo what they call the manufacturer on-tip chromatography. Finally, we also have partnered with Science Nova to automate their express dialyzer solution, which allow us to do plate buffer exchanges. These are some of the tools, these are some of the modules that we have that are suitable for LC-MS, but of course we have many others. I would just like to clarify a little bit better what IVD means for Hamilton instruments. So as we all know, Europe will dramatically increase the regulatory oversight of diagnostic products from 2020 onwards. We will go from IVD directive to IVD regulations. And this is expected to have a significant impact on the way that clinical laboratories work in the future. And this is particularly important for laboratories doing LC-MS analysis, given the fact that the majority develop and validate their own assays, what we call LDTs, homebrew assays, which do not require a CE-IVD mark yet. From 2022, only LDTs for which an equivalent CE-IVD test does not exist, meaning that there's no commercial kit available, will be permitted for use. So having said that, just to clarify here, maybe on the bottom, fully IVDD or IVDR certified solutions must include the instrument, the reagents, and the method. So it's a whole package. That means that the instrument needs to be classified and certified, Hamilton instruments classified and certified partner reagents with method on the IVDR. This means that we in Hamilton can only offer a fully, cannot provide a fully certified system. Currently, we only offer the IVDD compliance solutions together with current systems. And we are working in solutions that are IVDR. I just wanted to make clear that it is for IVDR and IVDD solutions, it's not only our instruments that has to be certified and classified, it's also the reagents from our partner and the method. This, I would like to invite you to have a look at the content that we have recently developed around the topic of LC-MS for drugs of abuse testing. We have here an ebook which explains the different types of the drugs of abuse testing and how LC-MS plays a central role in it. We have also a case study that was done with Labo Berlin, more specifically with Lars, where he goes into detail about the method of drugs of abuse testing in oral fluid samples. And finally, this webinar that will be available on demand after today. Beyond that, we have also other ebooks and in LC-MS. You might be interested in looking at those too. We have one in LC-MS for therapeutic drug monitoring, and we will be launching one in October for LC-MS in biopharmaceutical development and biopharmaceutical research. With that, I want to thank you. And I want to move straight to the Q&A session so we don't go straight with the, I see we have a couple of questions here. I would like to invite Nikolai and Lars again. Please unmute yourself and show your faces. And let me go first to the first questions. Then the first question here is if we can program the user can program or on their own. Maybe Lars can say something about it. Did you have to program on your own?
Yeah, so you can program methods on your own, and actually most of the method that we use for the oral fluid was programmed in-house by colleagues of mine. However, I have to say that you can't do it just like that. So I think Hamilton offers basic and advanced training in the Venus software, which is the software that runs the instrument. And if you want to program your own methods without using the help of Hamilton, I would recommend using these trainings. I'm currently undergoing these trainings myself. They're pretty good, I have to say. But yeah, because it's a very flexible instrument, and flexible also means it's complex to program. So yes, you can, but you need training beforehand. That would be my answer to that.
Thank you, Lars. And maybe Nikolai, you want to add something on that?
Right, okay. Yeah, what Lars said, I think is pretty accurate. After training, which you can book at Hamilton, you should be able to start programming the method straight away, but it requires, as I said, a bit of training beforehand.
Perfect, thank you. Another question that we have here is, have you had any issues with mucus when pipetting the saliva samples?
That's actually a very good question. So the short answer is yes, that can be an issue, of course, with saliva samples. What we do beforehand is that we centrifuge all of the samples, not with the Hamilton, just offline in a big laboratory centrifuge at maximum settings, just to sort of get all of the thick mucus to the bottom. But nevertheless, sometimes you have a sample that still has some mucus in it that gives you clots, not necessarily during the pipetting, but during the SPE process, because the microelution plates are quite narrow. And what we found, what helps then is to further dilute the sample with water or with buffer. Of course, you sacrifice a little bit of sensitivity for that, but yeah, sometimes that is necessary.
Thank you, Lars. I think one question that is maybe a little bit linked to this is, do you use TADEM to be sure that your pipetting has been okay? Maybe Nikolai, I don't know if you want to maybe explain a little bit what TADEM is?
Well, TADEM is basically a method, just respiration and dispense monitoring, which helps you to make sure that the pipetting worked properly. You can use it for monitoring if the pipetting was actually successful.
Okay, thank you. And Lars, you want to add something to that?
Well, the short answer is no, we don't use TADEM because we don't have the TADEM module installed in our instrument, so we can't use it.
Another question that we have also with problems is, have you had any problem with extreme high analyte concentrations, possibly residuum from peels?
So far, no, we haven't seen anything like that.
Short and to the point. I like it. Another one here is, are you able to run less than 96 samples at a time, or do you batch? If less than 96 samples, how do you keep the pressure of the vacuum stable?
That's actually a very good question. So if we don't have enough samples to fill the 96 well plate completely, we actually have programmed the method to fill the remaining positions with water. So we always keep the vacuum on the vacuum station for the SPE process. However, I would recommend running as many samples as possible, because once you use the SPE plate, you have to throw it away afterwards, because even the unused positions are filled with water, and yeah, we don't really have tested whether that affects their extraction capabilities. And these extraction plates are quite expensive, so I think that the one we use is about 400 euros a piece. So you pay those 400 euros whether you process 96 samples at once on that, or whether you process 2 samples. So of course, if you process a low number of samples, your preparation costs will rise quite significantly. However, I think there's a system offered by Agilent that also has a 96 well format, but where you can put small cartridges on single positions, so you can only use the number of or the amount of SPE material that is actually necessary for the number of samples that you want to process. But I don't have any practical experience with that yet, but we're looking into that.
Thank you, Lars. We have another question is, would you suggest using the vacuum manifold or the MP tools for SPE workflows? Which one would you suggest using?
I think that's a question for me, because as far as I remember, Lars has never worked with the MPE square. The two
No, I haven't.
Basically, which we offer, the standard systems. The first one is the Chrome Systems Master. Second one is the Dual Master, and both actually do not come with the MPE square. So if you want to use, they do come optionally with a crystal vacuum station. So if you want to use the vacuum station, you can do so. For SPE, it's as I said, it's a vacuum station where you create negative pressure to suck through the liquid through the SPE columns, or the SPE plate actually. Whereas the MPE square is for positive pressure, but this is not as a standard solution on the platforms. It would need to be integrated as a customized solution.
Great, thank you, Nikolai. There is a question here regarding the Venus training, so the training for programming. Could you explain a little bit, Nikolai, maybe, or maybe Lars as well, how is this training and how can people sign up for it?
Well, you can, I think I can answer this question. You can sign up for the training on our website. You can see what slots are still available. There's training run throughout the year. At the moment, as far as I remember, it is online, but we plan to have hands-on training as well in the future again. So the training goes, I think, over 4 days. It covers all the basic of the Venus software. If you have questions regarding the price, you should be able to contact your local sales rep. They can give you an exact price in your country. Okay, maybe Lars, you can comment on how you'd like the training.
Yes, I can. So so far I've taken the basic training. And yeah, I must say it was a pretty good experience. It was 4 days indeed. I did it online. Yeah, given the ongoing pandemic, everything's online. Although I would usually prefer to do it hands-on on site, because yeah, it's always nicer to work with the instrument itself. But it actually worked quite well in the online training. It's small groups, so if you have problems, you can interact directly with the trainer. There's a lot of practical exercises to really practice programming methods in the Venus software, and I actually learned quite a lot from that.
Great, that's fantastic. And I think that was our last question. We have one more here, but I think we will answer this afterwards. We just reaching the 5 minutes here. So with that, I really just want to thank Lars. I want to say Nikolai as well for the Q&A session. It was a very interesting webinar. And of course, you are able to download or to watch this webinar on demand, as I said. I would also recommend you to see the case study that was written for this particular user case. And if you have any questions, don't hesitate to send an email. We will come back to you. Thank you for attending the webinar and stay safe.